I do have your scantrons and exams here. I wanted to go over a couple of things really quickly beforehand. Some of this was more, oh, sorry, I lost a little bit on the side there. The pseudosymmetry, not pseudo-equivalence. We had some issues with that. I guess I didn't do a terribly good job of explaining it. Not really critical. We'll talk about adenovirus again later. Second one is what the tip of the drill did on T4. It's actually an enzyme and will degrade the inner membrane. Again, not terribly critical, but these are the questions that more than 80% of the people missed, which I think means I did a bad job of explaining them. On the other hand, this one's much more important, so I wanted to spend a couple of minutes talking about it. Which of the following is the most common mechanism we've discussed that makes virus capsid metastable? This is the whole idea of your capsid, and I finally got my printout of one of them here today, 3D printed version of a plant virus we'll be talking about a little bit later. Many, not all of them, but many of the cases where you have the problem, which is outside the cell, the virion has to be stable, but it still has to be able to release its genome. This is the whole idea of metastability, stable under some conditions, not stable under other conditions. And for the vast majority of the viruses we've talked about so far, and actually for the rest of the term we'll talk about, it's proteolysis which really does that. So you can assemble a nice stable particle before you do proteolysis, and then after proteolysis that allows usually a conformational change then to take place, which would have been another option but wasn't on the list. So you can release the genome under appropriate conditions. So that is an important aspect here that I wanted to emphasize, and obviously I didn't do a good enough job the first time around as far as that was concerned. The last one was everybody's favorite, that extra page at the end, and that's partly why it took me longer to get the exams done, because it takes longer to grade. Most people got this pretty well. Important is here, y-axis is your PFUs. It's really important that we're looking at infectious virions here, not virus, nucleic acid, etc. Time, of course, on the x-axis. You have a flat period. That's your latent period. Your latent period is up to here. After you go beyond this point, it's the end of the latent period. It goes up and it flattens off. It doesn't come back down. And so a couple of people had some issues with that. The next question had to do with where, the next part of the question, I should say, is where in this growth curve do you have the replication proteins being made? It's early on in the latent period. I gave people points for pretty much all the way through here. If it was over here, though, that's not the case. Or even right here. It's got to be before, because you need to make your virus genome. And so you have to be making the proteins to make that virus genome earlier on. So that's the exam. People have more questions about it. Please feel free to email me unless they're really quick.
I probably got that wrong. But on mine, I put it right at the end of the flat line because I figured that if they were made earlier, they would have replicated faster.
No, so it's the proteins you need to replicate your genome. So unless you have new genomes, you're never get. Here's an infectious particle. You've already got genomes. You've got everything you absolutely need when it starts to come up here. Yeah?
With T4 bacteriophage, how does the tip of the drill get through the outermost membrane?
We can talk about that more later. That's the injection process. But again, it's not that critical for this course. This was something I wanted to mention because many people had missed it. OK, finishing up with Lambda. I'm probably not going to have a chance to show the really cool video, but as you may notice, it's got 2-hour loan on it, so I'll take it over to the library and you can take a look at it. Really nice review, particularly for people who are really visually inclined, thinking about regulation and all of the extra steps that go along with that. Since I'm not showing it in class, stuff that's in there is not specifically going to be on the next midterm. Pardon? Oh, the name is Phage Lambda Foundations of Molecular Genetics by MobiFlix. It's a group based out of, I think, Irvine, who have done a couple of these molecular biology animations. But it's really quite nice. So what we talked about was transcriptional regulation last time, talked about lysogeny, a little bit about integration. What we haven't talked about is replication. So these are some of those other proteins that may come up. You may see them on some of the old exams if you're preparing for the next midterm. The only two important proteins as far as we're concerned are the origin binding protein, which is the O protein, and this is the origin of replication, and then all the rest of the machinery is going to be cellular with the exception of the P protein. And it's a really standard, bidirectional, semi-conservative, semi-discontinuous replication process. And particularly that's true in the early stage of replication, and Lambda is a little different than a lot of other bacteriophages. It's got two different stages of replication, although we saw that before for what other phage? Different stages of replication, the genome? 5x174. Unfortunately, we don't have too many other viruses to compare to yet. So yes, it's 5x174 because they have your double strand that makes more double strand, and then it switches over to making single strand. In the case of Lambda, you start out doing theta replication, and theta replication is just like you have with the E. coli origin of replication. Start at one point, go bidirectionally around the genome until you get to the opposite end. Of course, it's circular, so it's just the opposite place relative to the origin. On the other hand, late replication, and that's due to some expressions on the late proteins that we won't get into the details for, does rolling circle. And I just wanted to sort of diagram that here. So as this circle goes around and around, the DNA polymerases make these one really long piece, and that is multiple genomes right next to each other. And so that's the whole idea of a concatemer. And these concatemers, again, it's one big long piece. It's got multiple copies of the genome. Each of those individual pieces has to be put into the capsid. And if we can get this to go, let's see. Try and do this animation. Yeah, Ian.
DNAB, that's a bacterial origin?
Yeah, it's a bacterial. That's the helicase. It's the replicative helicase. So as always with replication, the key is pulling those two strands apart. So it's the helicase, which is the really important one. So DNAB is that helicase. Okay, so this is the rolling circle mechanism of replication here.
Conjugation and the reproduction of certain viruses, DNA is replicated by the rolling circle.
We don't need that. So here, rolling circle mechanism, you have this nicking enzyme, and all that does is it cuts the phosphodiester backbone and provides a 3' OH. This is supposed to be representing the new nucleotides being added here, and as you're replicating, it gives off an extra copy of the genome here. In the case of 5x174, this is just a single-stranded piece that gets religated. Here, you have semi-discontinuous replication that happens on, as it were, the lagging strand. Everything which is coming off of here, your rolling circle, is giving you the lagging strand. So you end up with now double-stranded DNA from this rolling circle. But that second strand is all cellular, quote-unquote, lagging strand polymerase. And hopefully you understand this just keeps going and going and going and going and going. So you end up with multiple copies of these genomes hooked up one next to each other. And again, those are concatemers. Yeah?
Didn't the polymerase actually methylate the part of the bacterial genome that it's already replicated so that it doesn't continue going around the circle? How does the virus overcome that?
Yeah, I'm not. Oh, you're talking about licensing. So once you've replicated, you don't start again at that particular position. Replication is happening so quickly here because it's only a 50 kilobase pair genome, which is smaller than that for Lambda, that there's no time for that methylase to come back in and remethylate. If you're talking about the E. coli genome, it's a 5 megabase pair genome, a 5 million base pair genome. So by the time the polymerases get to the other end before they restart, then you have time to methylate. Okay, so now you have this concatemer. Here, sorry, let's move over here. Do my pointer. The pointer's not working. Okay, so we've got a concatemeric set of genomes, again, one right next to each other, and each of those, of course, has a cos site in it. Cos sites are what? The cohesive ends. When the genome comes out of the virion into the cell, what is it? What form does it have? It's a linear form, and what's the end of that linear form? It's a cohesive end. And so these cohesive end sites, let me try and get my pointer here again. At these cohesive end sites, we have binding by the so-called terminase enzyme. Also, GP blah, blah, blah. We're not going to call whatever GP it is. It's just the terminase as far as we're concerned. So this terminase will bind to the cos sites and cut at those cos sites, leaving a cohesive end such that it can bind to the next cohesive end when it puts that into the cell. This terminase now goes to an intermediate in the structure, so a prohead, just like the procapsids that we talked about before for FIX174, for T7, etc. Binds to one position on the prohead, and this is the so-called portal protein. So terminase, portal protein. Those are the important ones to remember. So terminase binding to one end of the genome, cutting it, finding the portal protein. And what the portal protein does is it pumps the DNA inside the capsid. So here, with the hydrolysis of lots of ATP, it pumps in one genome and exactly one genome because the terminase is sitting here. It's lost its first cos site because that's been packaged into the prohead. Once it gets to the next cos site, what happens? The terminase cuts and goes and finds another prohead. And this whole process repeats again and again and again. This prohead is made, again, extremely similarly to what happens with FIX174, only now the scaffolding protein is just on the inside, not on the outside, and requires a particular protein that should sound vaguely familiar. GRO-E. Where do we know GRO-E from? Last term. Chaperone, exactly. So all the chaperones, HSP60, etc. How were these found? They were found in mutants that didn't grow lambda. So that's why they're called GRO. It's for lambda growth. Has nothing to do with chaperones, etc. These were found as mutants that did not allow lambda growth. So we found out later they were chaperones, probably important for folding lots of proteins. But it turns out that they are just as important for putting together proheads. So making sure that these proteins are folding properly. Looks as if the lambda head, so the actual icosahedral unit, is also cross-linked, like for HK97, with these interdigitated cross-links. We don't have a high enough resolution structure to know that yet, but that looks like what's going on there. So we have a head, we've got the DNA package inside of it. What do we need now? We need the tail. And this tail is really kind of fascinating. There's a protein called the tape measure protein. And the tape measure protein, turns out, is exactly like what it sounds. It's a long protein that determines exactly the length of the tail. And if you shorten the tape measure protein, you have a shorter tail. You lengthen the tape measure protein, you have a longer tail. All of which still seem to be perfectly functional. So it's a really interesting way of getting this tail structure. And unlike the case for bacteriophage T4, where it's helical, you remember that helix, which is the one that compresses, that was wrong in the video because it was the wrong-handedness? That then is just rings here. That's the T4 is a helical. This is just rings. These are just individual rings of proteins that go along this tail structure. and the number of rings is determined by the length of this tape measure protein. It's also the largest protein in the genome. Don't have a chance to take a look at that. So we finally got our virions. How do they get out? Turns out that this is, again, very similar to what we've talked about before. There's a peptidase that breaks down the peptidoglycan. There's a holon that forms in the membrane, and it escapes, basically making that hole. And just like we've seen before, it's the timing which is really important here. You build up enough of these holon proteins in order to make a hole. If you make too much of the holon protein too early, you release too many unfinished particles. If you wait too long with this, you end up really increasing your burst size, which may or may not turn out to be useful for replication. And so it appears that evolutionarily, the expression of the holon is really tightly coupled to expression of fully formed virus particles.
What would be bad about too big of a burst size?
What would be bad about too big of a burst size? The problem there actually seems to be having to do with the assembly process, is that you end up making lots of non-functional versions of your virions, probably because you're just trying to make too many of them. But that's a open question, and you could also argue, depending on your population, how many of the other phage are there. In fact, the meeting that I went to on Tuesday in Arizona, they were talking about evolution of these various different mechanisms in Lambda. So send me an email, I'll send you some more information on it. Fascinating stuff, but way beyond the scope of this course. More questions on Lambda?
Sorry, so too big of burst size is it's just not efficient enough is the main problem with it when it's too big?
Yeah, so it seems to be that it's more of an efficiency problem. Is you don't have, you're going to have a lot of non-functional particles in that case, and probably you would end up killing the whole population of E. coli around you in such a way that you couldn't continue to maintain your population. But that's a lot of hand waving. It's that evolutionary stuff. More questions on Lambda? What does he mean? Time to get out your clickers. So, genome replication by Lambda is most similar to genome replication by Q beta, 5X174, bacteriophage C7, HIV1, E. coli. Let's modify this so it should be late genome replication. Let's make it a little clearer.
Now you don't have to remember the notes.
100%? Five. And lo and behold, we have 100%, and we're right. Yay! Never can be sure with Stedman, you know. Yeah, basically, again, we've covered Lambda here, probably in more detail than we needed to, but we could spend a whole term talking about Lambda, and it would be great. Now, even worse is we're going to spend about 45 minutes talking about all plant viruses, which is absolutely ridiculous. Again, we could have a whole course talking about plant viruses. So, of course, plant viruses, we're skipping a whole bunch of stuff here. And just by reference, this is the book on TMV, or a book on tobacco mosaic virus. And we'll spend, I think, 2 slides on tobacco mosaic virus here. So a couple of important concepts here. Most of these, and I didn't bring my virus taxonomy book because it's too darn heavy, but probably the majority of different virus species in there are in fact plant viruses. Highly diverse, almost all of them RNA viruses, and the vast majority positive strand RNA viruses. So we talked about MS2, Q beta in terms of the RNA bacteriophage. For the next couple of weeks, we'll be talking about the RNA viruses that are infecting eukaryotic cells. One of the examples here, and in fact one of the best known of these, is one that's called the, bromovirus family. Why is it called the bromovirus family? Because the broccoli mosaic virus family. Plant virologists have a tendency to name their viruses based on what they infect and what kind of phenotype they see. Now, it turns out the best understood of the bromoviruses is cucumbervirus, because it's a cucumber mosaic virus. So, but that is not as critical as the concept, which is a really bizarre one, about many of these plant viruses. They have segmented genomes. And we talked a little bit about segmented genomes already. I like to think of those as sort of individual chromosomes. And so they need multiple chromosomes in order to be able to replicate. But what's really bizarre about a lot of these plant viruses is that each of these capsids contains one of the chromosomes, but they need multiple chromosomes in order to be able to replicate. So they actually have to be infected by multiple capsids containing multiple genomes to be able to actually functionally reproduce. And evolutionarily, how the heck this happened? Again, very open question. There are a couple of other things about plant viruses which are really pretty specific to plants. Plants do have circulatory systems where they move liquids through the plant, but usually the plant cells are really quite isolated one from another. And because plants can't run away from herbivores and all of these other things, they have lots of defense mechanisms. So it turns out that almost all plant viruses have something that's called a movement protein, which allows those virions or the virus genome to move from cell to cell. Yeah?
Is there cooperativity with the binding of, because you said you need multiple capsids, so is there like, if you infect with one capsid, you're more likely to be infected with like other capsids?
So we'll talk a little bit more about that later, is how you get this kind of infection. It has to do with actually the fact that plants are really tough and can't run away, and so these viruses can't get in by themselves. They need help, and lots of help. So, and then briefly at the end we'll talk about tobacco mosaic virus. So again, some of the key concepts here: almost all RNA viruses, many of them have these segmented genomes. Why? Probably because of just the fact that plants are very different on a cellular level than most animals and certainly bacteria. A number of these virions undergo some really fascinating conformational changes, and that's what I showed you on that first slide. Interesting host effects. And then there's this really fascinating example that I forget if I've talked about here before, but this is really an example of a good virus. And so as far as the plant's concerned, it's actually much better off if it's associated with this particular virus, also called the thermal tolerance virus. And then we'll look at a little bit at the helical structures of TMV right at the end. So CucumberMosaic virus. Turns out that CucumberMosaic virus is sort of the classic, best studied of these viruses, and most of the chapter in your textbook talks about CucumberMosaic virus. So we'll talk about where it came from, what it looks like, how does it get in, then the replication and packaging thereof. Oh, actually, I deleted this thing on the satellite, so you can chalk that out of your notes. And then we'll talk a little bit about cowpea chlorotic mosaic virus. What does it infect? Cowpeas. What does it make? Chlorotic mosaic phenotypes. But mostly because this is really well understood in terms of the structural changes that happen in the virion. Hot viruses, best kind of viruses. And then TMV again right at the end. So, sorry to keep beating on this particular case, but we're talking about mostly single-stranded RNA viruses with a positive sense. So these just have to release their RNA. Once that RNA gets inside the cell, the cellular machinery will translate it. And one of the things that it has to translate, of course, is a viral RNA-dependent RNA polymerase, because you've got to make more of that particular RNA. And most of this, it turns out, happens in the cytoplasm. You don't actually need to go to the nucleus for the vast majority of these plant viruses. Where did CucumberMosaic virus come from? Yeah, let's see what happens. So I had to look and try and find it, because this is a happy cucumber. This is an unhappy cucumber, which has been infected by this virus. And what happens is it's not the whole cucumber, it's just bits and pieces. And so that's what the whole mosaic business is about here. Found relatively early on in terms of virus studies, way before any of the electron microscopes, etc. Pretty soon after bacteriophage was discovered for the first time or discovered for the second time. So actually, I wonder if there was a party last year, 100 years of CucumberMosaic virus. But again, it's just one of these things that's causing disease after you put it through a filter. And so that was the original definition of this. And they push in the book chapter that's got a very wide host range. In fact, the widest host range of pretty much any virus anybody knows about. They're all pretty closely related plants. And if you think about it from the whole molecular point of view, all plants are represented by this tiny little twig down here relative to the rest of molecular diversity. So they're clearly focused on plants when they're talking about very wide host ranges here. Yeah?
So when you say infects in bits and pieces, does that mean each little area of infection needs to be from a separate, series of, virus capsids, that infect?
So I'm going to paraphrase your question and say, why do you have this mosaic phenotype? Why is it not a complete phenotype all the way across the organism or all the way across the plant? Probably actually has more to do with plant defense mechanisms rather than into multiple infections. You can, in fact, do a single infection in a single place and still get this mosaic phenotype, and that's because of those movement proteins that we talked about, and we'll talk a little bit more about in a little bit more detail here. Yeah?
So if you infect like the cucumber one on one side and one on the other, since it needs two will meet up.
Okay, we'll get to this whole multiple infection thing in just a second. So that was, I think, basically your question. Yeah, how do you end up getting these things? First, however, I want to talk a little bit about the structure. This is, as I mentioned before, a 3D printed model of a plant virus. Afterwards, you can come up and check, after you found your exams, what the T number of this particular particle is. It's actually slightly different than the T number of this one down here. It's a single beta barrel. And this is unlike the case for STIV and all these other ones that sound like they're potentially derived from one common ancestor way back when. Again, diversity-wise, this is what makes up the vast majority of the structure. There's a little bit of a projection that comes on the outside. And then most important, as far as these viruses are concerned, is this N-terminus here that's loaded with Rs. What does R stand for? Arginine. What's the charge of arginine? Positive. What does it interact with? Or, in this case, RNAs. So nucleic acids. So the nucleic acid interaction is all going on down here through this highly charged, highly positively charged helix. And that sits in the middle of this virion, which is a purely quasi-equivalent T = 3 icosahedron. Here's a 5-fold axis of symmetry. There's a 5-fold axis of symmetry. Here are your 3-folds in between. So hop 1, hop 1. So those are your structures. Now we're finally getting this whole business about multiple capsids. So plant cell walls, great protective mechanisms against the rest of the outside world. So how the heck do you get your tiny little virion into these kinds of cells? And the answer is by vectors. We haven't talked about vectors so far yet. But this is the nasty vector which transmits the vast majority of plant viruses, and particularly these cucumboviruses, are the aphids. Now, aphids actually only will feed on certain numbers of plants, but it turns out that they will go and check out a whole bunch of different kinds of plants. And they may look really small. Yes, this is a scanning electron micrograph, but relative to a virion, this of course is huge. And it turns out that the mouth parts of these aphids are completely loaded with virions. So when you have a virus infection in a plant, you have extremely large numbers of virions of all different kinds, packaging all different kinds of your segmented genomes in every given cell. So if the aphid comes in and literally chews a hole, so that's how the virus gets into the cell, is because the aphid has chewed a hole into the cell, and its mouth parts have massive numbers of viruses that are associated with it. And it's literally, when I say massive numbers, it's 10 to the 6, 10 to the 7 individual virions. So it's very, very, very large. And if they bite into, and they're actually not inoculated, a cell that already has these virus particles in it, then those will get stuck to the aphid's mouth parts. One thing that you would expect, if this is the case, that you'll have very, very, very high titers of virions in any infected cell. And this is exactly true. It turns out that plant viruses, when they replicate, make ridiculously large numbers of virions that can then get picked up by these aphids. So the entry process here really has to do with damage to the plant. And anyone here worked on plant viruses? No. So one of the standard ways to actually inoculate a plant is to take a leaf and scrape it a little bit with a razor blade or whatever, and then just put your virus on it. So you're just pretending to be an aphid. And literally that you can get massive amounts of virus production that way. So it's literally just the damage. And the damage is usually, of course, for plants, it's some kind of insect which is coming in. And then, because you've got so many of these individual particles that are associated with that damage, or in the case of the aphid, then just statistically speaking, you are going to have enough of these individual genomes in order to get a productive infection, which again seems really bizarre from a evolutionary point of view, but it works. So what's in there? In the case of CucumberMosaic virus, you actually need 3 different virions with 3 different sets of genomes in them. Also very conveniently called RNA 1, 2, and 3 and 4. Okay, great. No. Perfect names, no, he's not going to expect you to remember all of them. But they encode, not surprisingly, the important things that you need for virus replication, which are what? Okay, what do you need? Capsid, yes. What else? Replicase, RNA-dependent RNA polymerase. And what I say about these plant viruses? Movement proteins. It's basically all you really need. There's some other modifications that happen here, but that's really, for the most part, the only real 3 things that you need. And they have here, conveniently, it's the CP protein. Okay, so we've got our capsid protein already. RNA-dependent RNA polymerase is up here. And then the movement protein turns out to be this 3A protein. These RNAs, and when we talk about our RNA viruses, we're going to continue to talk about particularly what happens at the 5' end and at the 3' ends of these genomes. What the normal messenger RNAs have. They've got, at the 5' end, they have a cap. Yes, thank you. It's Friday. You guys will be awake. And at the 3' end, what do they usually have? Poly-A tails. So these guys have caps. They've all got normal caps that are present at the 5' end of the genome. How they get their caps is an interesting story. On the other hand, at the 3' end, they don't have a poly-A tail. Now, why wouldn't they necessarily want to have a poly-A tail? What are poly-A tails good for?
Protecting the RNA.
Protecting the RNA by doing what?
Not letting the nucleases.
Not letting the nasty exonucleases chew in at the end of your genome. So the way that these plant viruses deal with that is that they have this secondary structure, hairpin structure, very close to the 3' end of their genome. We'll take a look at that in just a second. So that's what these individual pieces look like. Turns out that if you look at the length of these individual pieces, this one's about 3,000 nucleotides. This one's about 3,000 nucleotides. And these two together turn out to be about 3,000 nucleotides. Give you one guess. How many nucleotides fit into a CMV capsid? 3,000. About 3,000, exactly. So that seems to be the evolutionary drive for having multiple capsids. So you just have one capsid protein, makes this nice T equals 3 icosahedra, packages 3,000 nucleotides. So that presumably now is a selective advantage, is just to have that one capsid protein in this one set of 3,000 nucleotides. But what that means, of course, is that we need to have multiple capsids in order to get a full infection process. Yeah?
Are all these capsids distinct for the genome segment that they are packaging? Or is it just kind of we make a capsid and we throw whatever segment of genome we have in there?
Okay, so what would you guess based on what I've told you? All right, so actually I'll repeat Ian's question here for those of you who didn't hear. So is each of the capsids specific? So you have a capsid for RNA1, you have a different capsid for RNA2, and you have a different capsid for RNAs 3 and 4.
They're identical.
They're identical to each other. So what does that mean about packaging was the other question. So it's whatever piece that's about the right length, so 3,000 nucleotides. So 3,000 nucleotides, boom, package. 3,000 nucleotides, boom, package. So that's, again, evolutionarily, it works. Yeah?
Does that then mean that you get random amounts of, for each infection or each replication cycle of whatever segment? It's not going to be just kind of you get this percent of this segment, this percent of this segment, this percent of this segment every time?
Yeah, so the question basically has to do with packaging efficiency, which is a different way of putting this. It seems to be if you have your population of random virions, on average, you'll have 1/3 that have RNA1, 1/3 that have RNA2, and 1/3 that have RNA3 and 4. But you can do the Poisson distribution and figure out how many particles you would have to have in order to have a productive infection. And I won't force you to do that, don't worry. But I will ask you this next question for clickers. So here, let's go for another 100%. Oops, if I turn it on, that might help. What's the minimum number of different capsids required for successful infection by CMV? Come on, let's go. Yes, finally. So the capsid with genome. So I should actually literally say different virions rather than different capsids, because that would be protein plus nucleic acid.
So like that phenotypes would like jazz together for different capsids? Is that what you're saying?
My brain is very slowly.
I feel like some of the virus produces so much of itself to help it, but that would also
5.
reduce the chances that a beneficial mutation would actually, because you have so many particles,
Okay, we've got slight differences of opinion here. But yeah, the minimum number of different capsids plus RNA, it has to be 3. Now, usually it's have to be way more than that, again, because of the Poisson distribution. But the absolute minimum would be 3. Speaking of 3, what about this 3' end of the genome? So I mentioned it's tRNA-like. Well, if you draw out the secondary structure, this to me doesn't look like a tRNA at all. Here's the 3' end. What it does is forms this really interesting secondary structure. And this is the same, actually, at the 3' end of each of those individual genome segments. So they all have this same sequence at the end of the genome. But even though this, to me, doesn't look like a tRNA at all, it fools the amino acyl tRNA synthetases and gets them to put an amino acid here at the very 3' end, just like you have with tRNAs. They've got the 3' OH. That's where you attach your amino acid that would be used for translation. So what does that mean about the 3' end of your RNA? It doesn't look yummy to the exonucleases anymore. So it's protected that end. So it's a modification, cellular modification, amino acyl tRNA synthetase, which puts the amino acid at the end. Yeah?
Is that adenine not extremely unstable there? The weird one that's just like, I'm going to bind to this site.
Oh, this one that's sticking out over here?
Why is it bound to this?
Why is it hanging out over there? Well, the problem is that this is a 2-dimensional representation of a 3-dimensional structure.
Still, it's skipping like 4 bases to get.
Well, no. So this is just again, it's to try and draw this in 2 dimensions as opposed to being a really 3-dimensional image. I should try and see if I can find. I think they actually have a pretty good idea what the structure of this actually looks like in 3 dimensions. But let's go back here real quickly. But the this connection here, so it's this A goes to that A. This is a normal phosphodiester with that particular length. They would just be completely overlapping in order of drawing it here.
I guess it's just the 3-dimensional structure.
Yeah, so the 3-dimensional structure is such that this is pretty well protected. Yeah?
So I just want to make sure it is forming this to protect the 3' hydroxyl so that nothing's coming in and trying to degrade it?
So apparently, again, this is a why question, we don't know, but it does appear that that protects against exonuclease activity, which would be degrading in at the 3' end of your RNA. And we do know, plant people work on these plant viruses, is that if you mess up this structure and you don't get amino acylation at this point, those RNAs are degraded really rapidly, and you don't get good virus replication. Yeah?
If you mess it up, you don't get the protein binding either. And that's what the corollary means.
I'm not sure I quite understand what you mean by the protein binding.
Could be misunderstanding. Yeah. A protein binds to the 3' end, and that's what protects it from
No, so it's an amino acid which gets added to the 3' OH. So it's just that amino acid that gets added to the 3' OH. And I don't remember which one it is.
Okay. But if you mess up the structure, you don't get the amino acid binding, and then you don't.
You won't get the amino acid covalently attached. So it's not. This is not a binding. This is just like what normally happens with tRNAs. You know, charged tRNAs with amino acyl tRNA synthetases. So that activity is exactly the same. So it's a covalent linkage of an amino acid to the 3' end of the genome. Okay, so what are these virus proteins? No, I don't expect you to remember all of them, but a couple of important ones. What do we say we needed? We need an RNA-dependent RNA polymerase. That's RNA2. The coat protein, RNA4. Okay, so those are the only 2 that we really absolutely need. What else? RNA1 is interesting. It has a RNA helicase domain, which is actually exactly what you would expect to need, because whenever you're doing RNA replication, or replication at all, you're going to end up with double-stranded pieces. And in order to make more of these single-stranded pieces, because that's what's getting packaged, 3,000 nucleotides each in each of your individual virions, you have to be separating these 2 strands. And so having an RNA helicase turns out to be very common in a lot of these other RNA viruses. There are these other 4A, 3 we'll talk a little bit more about later on. I would ignore these these other ones here. So RNA helicase, RNA-dependent RNA polymerase, and the coat protein. 3, again, we'll get back to in just a second, but you can guess what it is because it's that other one that all the plant viruses need. Movement protein, exactly. But first, I want to talk about replication. Replication here, again, uses this RNA helicase, which is encoded in RNA1. But these RNA viruses, actually just like all of the other RNA viruses in eukaryotic cells we're going to talk about, have a big problem. Yeah, yes, there's this RNA helicase, which you need to do to separate the two strands, but needing an RNA helicase means that you have what? You have double-stranded RNA. Double-stranded RNA is something that cells do not like at all. And so they've evolved multiple mechanisms, including RNA interference, including all of different kinds of interferon mechanisms. Double-stranded RNA is bad as far as the cell is concerned. And the reason for that is because it looks like an RNA virus which is trying to replicate itself. Again, not terribly surprising from that point of view. So these RNA viruses have to have ways of hiding from that system. And there are lots of different ways. We'll talk about them as we move through for these other different RNA viruses. The way that CucumberMosaic virus does this is what it does is it makes its own little organelle inside the cell and undergoes RNA replication in that particular part of the cell. And how it does that, it's this protein 1A, which in addition to having helicase activity, forms these little, basically internal organelles or vesicles inside the cell where replication takes place. So you have your RNA-dependent RNA polymerase, 1A, gets what else is called sequestered inside the cell, and then it's just this individual RNA which is being produced. Turns out that there are also translational factors that are really important for this. Initiation factor 3, which is just, as far as we're concerned, is just a translational initiation factor. Why are translation factors very often used in RNA virus replication? We talked about this for MS2, Q beta, because they love to bind to RNA. So they're RNA binding proteins. They're very common inside the cell. So it's not surprising a lot of these RNA viruses help out with this. And there's also a DNA J-like protein. What's DNA J do? So it's a bacterial protein that seems to be important also for protein folding, not unlike the GRO proteins as well. So these are the host proteins that are involved in this, and then the RNA helicase comes from protein 1A. The other thing, sort of maybe as a aside as far as this is concerned, many of the proteins in these RNA viruses, let's just back up to that table, have multiple activities. And so this is a nice example. If you just look at RNA1, has an RNA helicase domain, but also has a methyltransferase domain. Why the heck would you need a methyltransferase domain? Where do you need methyl groups on your RNA? The 5' cap is all about methylation. So this is a capping enzyme, a sequestering enzyme, and an RNA helicase. So all three of these are what's encoded in RNA1, which is made basically as one big protein. So what haven't we talked about? Movement proteins. Oh, sorry.
I have a question about the replication.
Yeah.
Inside that little organelle, is it one replication or are there multiple replications occurring inside the organelle?
So the question is, are there multiple genomes that are getting replicated inside one organelle? So each of those different segments, or is it one segment per organelle? I have no idea, but I'll try and look it up. It's a great question. I don't know. I would guess one, but that's a guess with very little basis for actually really understanding. So what we haven't talked about are the movement proteins. It's a 3A protein in CucumberMosaic virus. It's important for this. And basically what these proteins do is they'll bind to individual genome segments and get them across these plasmodesmata, which are between cell to cell. So your aphid has chewed into this cell, released a bunch of virions here. Some of those will get translated and make 3A, and then these will all be transported to these cell-cell connection points. Once they get from each of these cell to cell, eventually they'll get to the circulatory system of the plants, and you people probably know more about this than I do because I had 1 biology course as an undergraduate, that then allow these genomes usually to get transported through the rest of the cell. And there are really sort of 2 steps in terms of movement in these plant viruses. The first one is this cell-to-cell movement, and then once it gets to the circulatory system, moving throughout the entire plant. And so that's where it seems that you end up getting this mosaic phenotype, and you have some cases where this kind of cell-cell transport can take place, but you can't get this long-distance transport inside the plant taking place. But in other cases, you do have that, and it just seems to be the plant immune responses, which we don't have time to get into today, called the hypersensitive response, et cetera. So that's all that I wanted to talk about in terms of the CucumberMosaic viruses, the cucumoviruses. Now I want to spend a couple of minutes talking about some really cool other things that people have figured out what to do with plant viruses and plant virions. So no more questions actually on this replication process. Yeah?
The movement, the 3A
Yeah.
movement protein, and is that a motor protein? Is it moving along the...
So the question is how does 3A work and how do movement proteins work? The answer is I don't know completely, but what I do know is that they actually mostly just seem to be protein-protein interactions with things that you have at the plasmodesmata. So they're not motors per se, and they don't seem to be associated with the cytoskeleton, at least not to my knowledge. But it's mostly just a protein-protein interactions, again, particularly for the plasmodesmata. OK, so this virion that is in all of my introductory slides, why the heck am I bothering with it? Well, because it's this beautiful T equals 3 icosahedron. No, that's not the reason that I'm talking about it. The main reason is that over a number of years, and this is true, in fact, for a lot of plant viruses, but best understood for this cowpea mosaic virus, is that this capsid, with a change in pH or a change in ionic strength, which is exactly what happens once you get inside the cytoplasm, it expands and basically pulls apart. I don't know if you've seen those. I probably should have brought one of these. The little balls you can pull apart and make these huge changes. Basically, it's exactly the same thing that's happening at almost an atomic level. And what it does is it opens up these holes here. And these holes are conveniently just the right size for your genome to escape from. But it turns out that you can just, by expressing the capsid protein, in the absence of any RNA whatsoever, you can make these particles and reversibly open and close them. And this is really cool because it means you can put anything that's going to fit in one of those holes inside this virus particle. And because plant viruses make these ridiculously large number of virions, you can end up with very, very large amounts of these individual particles. So if you could figure out how to load different things inside these virus particles, say by changing that N-terminal helix with all the arginines on it, in order so it would bind something else, you can put all kinds of different things inside these particles and then do something with them. So what have people done with them? One of my favorites is actually taking together with these produced particles, loading them up with basically magnetic beads on the inside of each of these particles, which are now really, really, really tiny. And you can get all these virions, maybe a little bit hard to see here, but these virions all line up right next to each other. They're nice, packaged. In this case, you can get really nice monolayers of virions. You can blast away the organics and just end up with the metals. And if these are Magnetic, you can actually use these just like you would for some kind of storage array, like a hard drive, for instance. Magnetic changes, so storage purposes. And in fact, when I was working on this, the lab I was working with had this big contract with Panasonic to try and make really high-density hard drives, high-density storage processes. Another thing you can do with these is magnetic resonance imaging. One of the big problems with MRI is getting contrast, getting large amounts of metals associated with exactly where you want to image. So another way of doing that, you can put chemotherapy drugs on the inside of these particles, get them to open when you get into a tumor, or you can inject these virus particles right into the tumor directly, have them open up, release their chemotherapy drugs. Really fascinating bio-nanotechnology processes. A lot of this was pioneered by Mark Young and Trevor Douglas, who were at Montana State University and Indiana University. Disclosure here, I worked with Mark for a couple of years before I came here. Montana State University, they were really good at doing these plant viruses, but Montana State is also really close to Yellowstone National Park, which is where my love for these extreme viruses come from. And one of the examples that people studied there, and this was actually not the group that I was working with, but one literally right down the hall, was working with this really interesting plant called panic grass. Why is it called panic grass? It's called panic grass because it grows at about the highest temperature known for plants to actually be able to stably grow. This is Celsius, and this is, it's a little hard to see where the probe is here, but basically, here's the probe going into and measuring the temperature of the soil right around the roots of this plant, 50 degrees plus. These plants can survive under those conditions. It's really pretty amazing, and in fact, when you go out into any of these thermal areas and you see where the plants start to grow, that's where these guys are, and you can put a thermometer in the ground right there. It'll be 50 degrees C. And they're the only plants which can grow there. Curiously enough, this Dicanthelium lanuginosum, and no, I don't expect you to pronounce it or remember it, can only do this when it's infected with a fungus. Wow, that's pretty bizarre. So you get rid of the fungus, the plants can't grow at those high temperatures, actually die pretty quickly. So Curvularia fungus, this is a fungus right here. It lives inside the plant, so it's an endophytic fungus. But why is Stedman talking about this in a virology class? Let's see. Because it turns out that it's only if that fungus is infected with a virus does it provide the thermal tolerance to the plant. Wow. Yeah. Why? Evolution. Just absolutely amazing process. And these are the data, in fact, from that particular paper that was published in Science a few years ago now. So if you have your normal plant with the fungus, and the fungus is infected with a virus, it grows perfectly happily at 50 degrees. If, however, you don't have virus, it doesn't grow. If it doesn't have your fungus, it doesn't grow as well, and this is just your negative control here. So, reinfected, this works. No virus, really unhappy. No fungus, really unhappy.
Technically, that makes it a mycovirus then, right?
It's in fact a microvirus and not a plant virus. You are correct, yes.
Just to clarify, if it's just infected, if the plant is just infected with the virus, it still can grow, just not as well.
Okay, so this is the microvirus. So it's actually, the plant is not infected with the virus. It's the fungus which is infected with the virus. So it grows really, really badly under those conditions. But it grows perfectly happily if it's infected with the fungus which is infected with the virus. And this is, in fact, an overview of looking at this thing. You see, if it doesn't, it's got real problems. If, however, it's infected with both the fungus and the virus, this has a really nice growth. And this is from a really nice paper written by Marilyn Rusick, who's now at Penn State University, on what she calls the good viruses. Yeah?
Is the relationship also mutualistic between the fungus and the virus? Do they both benefit?
So does the fungus benefit from the virus? These fungi just basically only seem to grow endophytically, and so whether there's a huge benefit from that, I don't know. Okay, we'll continue, just finish up with TMV on Monday, and have a nice weekend.